All right, biology again. This is Unit 3. This is going to be video 3B, active transport. Now, active transport is a little different from passive transport because it uses extra energy. And this is where we use that energy that we get from things like carbs and lipids that we learned about in Unit 2. So active transport will move materials across a membrane, but it may go against the concentration gradient. So active transport doesn't necessarily care about the concentration, it cares about what we need to get into the cell. Okay? So here, passive transport, we talked about this. We were always going from high to low, okay? Moving from high to low, high to low. But active transport doesn't necessarily do that. Active transport gets us what we need, whether it goes with the concentration gradient or not. And it usually uses this little molecule we call ATP. It's where we get our energy from, where our energy is stored. You can see here the concentration gradient, this is the low one, so it should be going from high to low. But this little protein here is moving things from the low actually to the high. So it's moving uphill. So it needs extra energy to go uphill against the gradient. An example of that, let's see if I got the right one there. Yep, that's what I wanted. Sorry, guys. An example of that, you guys, is called endocytosis. It's where a cell actually will, this is a large cell, usually like a eukaryotic single cell. It'll actually wrap its membrane around something that it needs, and its cell membrane will take that into it and absorb it and make kind of almost like a little bubble around it. So the membrane will come around it, phospholipid bilayer will wrap around, seal itself on the outside, and then we move material into the cell. Okay, endo means in, cyto means cell, endocytosis means to bring something into the cell. Okay, examples of that, first one's called phagocytosis, and that's where the cell eats. So phagocytosis, you guys, is a cell eating or bringing in solids. So you can see right here, here's a solid. The cell is going to wrap its membrane around this material that it wants to take in. Then it creates a little vesicle or a wall around it, and then it'll seal up the outside again. And related to that, you guys, is called pinocytosis. Again, it's endocytosis taking into the cell, but it's taking in liquid. So cells need water, and if we're not getting, that water or that liquid that we need through the membrane, sometimes it might happen through the process of endocytosis. So here's a membrane wrapping around it, making a little vesicle, and bringing it into the cell. Okay. Now the opposite of that is called exocytosis. Exo means out, cyto means cell. It's where a vesicle inside the cell, sometimes lysosomes, remember those lysosomes that clean up our cell? Sometimes the lysosome will work on taking dead organelles and material and moving it and bonding with the membrane and then releasing them outside the cell. It's just a way for our cell to get rid of waste. Alrighty guys, so here's the first quick little video about passive and active transport so you can see an animation of it. Okay, let's talk about passive first. Alright, you got simple diffusion, facilitated diffusion, and osmosis. Membrane, here's some protein channels, phospholipid bilayer. And they're calling it a fluid lipid bilayer because it moves fluidly. Okay, it's very important that it be able to move and be flexible. Otherwise, we couldn't move and be flexible. When it's talking here about polar molecules can't move through the outside, that's water is a polar molecule, so that's why we want that hydrophilic on the outside. All right, let's take a peek at diffusion here. All right, we know it goes from high to low concentration. There you go, you guys know that. Remember, that's our concentration gradient. High concentration, low concentration. Okay. Just like going down a hill. Right. Now, diffusion is going to depend on what your concentration is, temperature, the charge on your molecules, and also how big the molecules are. All right, there's your membrane again. They're just going to show the concentration gradient moving from the inside to the outside. They're saying diameter, the size is small, so it's going to move quickly to the outside. And we met equilibrium. Good to go. All right, let's take a peek at facilitated. We're going to move through a protein channel here. Remember, this is your key in the lock. Not really going to take any extra energy. It's just going to move it right through the cell membrane. It's a form of passive transport. Again, from high concentration to low concentration. Not using energy. There's your proteins. That's your lock. And the key's going to come in and unlock that. And there it goes. Haha. Pretty fancy, right, guys? Facilitated diffusion. Okay, there you go. That's facilitated. Let's take a peek at osmosis. All right, these are passive transport, remember. You guys know those terms. Hypertonic, higher concentration, hypo, lower concentration. Water always wants to move from this into the cell or from the cell out to hypertonic. All right, let's look at our water. I'm going to kind of fast forward here a little bit, guys. Okay, oops, sorry, I'm going to go, I want you to see the osmosis one, sorry. Fast forwarded too much. Where are you, osmosis? There it is. You just saw those. Right. Hypotonic water is definitely going to move from here into the cell to the hypertonic. There it goes. That's osmosis. All right, so let's check out active transport really quick. Remember, this takes energy. This won't take long. All right, we're going to talk first about what we call an ion pump. There is one called the sodium-potassium pump that if you guys take microbiology in college, it's so complicated sometimes, but it can also be really cool to learn about. Remember, we're going against the concentration gradient. Here's my cell membrane. So I can actually move against the concentration gradient. I can move out from here, out of the cell. And that's active transport. It uses energy, and you can move against the concentration gradient. Okay. Ion pumps. And that's good. You guys are good on that one. All right, and I think we have one more thing. Hold on. All right, guys, quick little video. This shows you phagocytosis.
In phagocytosis,
it's going to show the cell actually wrapping around this bacteria and kind of eating it.
phagocytes are attracted to the area of invasion by chemical products of the microorganism, phospholipids released by injured mammalian cells, or by components of the complement system. The phagocyte moves into the area of invasion and then attaches to the microorganism. The attachment is mediated by a variety of surface receptors, including antibody, lipopolysaccharide, and complement receptors. For example, the C3b component of complement coats bacteria or other particles and then binds to C3b receptors on the phagocyte. This process of coating to enhance phagocytosis is called opsonization. The microorganism is then engulfed by the phagocyte into a vacuole known as a phagosome.
All right, that's all you guys have to know. You don't need to understand how they move together at those sites, but just so you guys can see how the single-celled organism can come and wrap around that particle, okay? All right, you guys have one more slide. It's called the endosymbiotic theory. Okay, this is the last slide in the unit, actually. This, you guys, is the idea that at one point in time, prokaryotes lived separately on their own, like small bacteria that probably resembled mitochondria and chloroplasts, and eventually they began living together, were wrapped around by a cell membrane, and became what we call eukaryotic cells. Okay? So that's the endosymbiotic theory, the idea that small prokaryotes came together to make eukaryotic cells. Because so far, our evidence that we have from evolution through fossils is that prokaryotes have been around for a much longer time than eukaryotes. So we think that our eukaryotic cells came from prokaryotes. Okay? That's the end of your notes. Congrats!